Hello, BSc second year students. Now, in today's online lecture, we will study the glyconeogenesis and citric acid cycle. So, on the board, this hub is for the citric acid cycle that we will study later on. First, we will study the glyconeogenesis. It is a biological process and it is also called as the neoglucogenesis. Remember, glyconeogenesis is also called as the neoglucogenesis. This is a bit. This is today's study is from the unit 3rd and carbohydrate metabolism is the chapter name. Remember what we are studying in the class online lecture. You remember unit 3rd, that is carbohydrate metabolism we are studying, and in carbohydrate metabolism there are different biological process so which occurs in the presence of different types of the enzymes. So in today's lecture, glyconeogenesis means neoglucogenesis we will study here, and here first we will study the definition of glyconeogenesis. So formation of glucose, listen here, formation of glucose or glycogen from non-carbohydrate precursor. Formation of glucose or formation of glycogen from non-carbohydrate precursors. When this carbohydrate is exhausted in the body, that time this process starts. So remember that. Now, non-carbohydrate precursors are, what is non-carbohydrate precursor? Question arises. So here is the answer. Non-carbohydrate precursors are lactic acid, amino acid and glycerol. Remember this. Try to understand. If at the time of the study, if you found any problems, any question arises, you can do the mobile contact. You can ask on the mobile. So, dear students, listen. Now, this biological process in glyconeogenesis, pyruvate undergoes the process and it converts into the glucose or glycogen. So here, pyruvate is undergoes in the biological reaction, biological process, and it converts into the glucose. So it is not directly in one step completed. So it passes through the several stages. So in the first step, this pyruvate is converted into the phosphoenol pyruvate. Remember, pyruvate is converted into the phosphoenol pyruvate, and this reaction is in the presence of a biological catalyst and enzyme. Then phosphoenol pyruvate is converted into fructose 1,6 diphosphate, and this reaction is the reversible reactions. Means this convert phosphoenol pyruvate converted into the fructose 1,6 diphosphate, and this also convert to the phosphoenol pyruvate. Here reversible symbol is shown here in the figure. There fructose 1,6 diphosphate is converted into the fructose 6-phosphate. Then this fructose 6-phosphate is converted into the glucose 6-phosphate. Again here the reaction is the reversible. And finally the glucose 6-phosphate is converted into the glucose. So remember, in every step there is the biological catalyst and enzyme is used. That enzyme do the conversion. In the presence of enzyme, a reaction is completed. So, dear students, this is about the glyconeogenesis, which is also called as the neoglucogenesis. Now, we will study the another biological process which is very important in the body, that is the citric acid cycle. This citric acid cycle discovered by the Kreb. So, after the Kreb, it is named by the Kreb. It is known by the Krebs. So, it is also called as the Kreb cycle. This citric acid cycle discovered by Kreb, H.A. Kreb. Here, remember it is discovered by the H.A. Kreb. This cycle is abbreviated by TCA cycle. TCA cycle, tricarboxylic acid cycle. Then the net gain of energy is in the form of ATP, and it is always 24 ATPs. Remember, 24 ATP energy is a net gain after a cycle. How the cycle starts? So here glycolysis is the basic. So glycolysis occurs and it forms the pyruvic acid. That pyruvic acid combines with the acetyl coenzyme A. Parallelly, another biological reactions are here. Fatty acid, after the beta oxidation, it gives the acetyl coenzyme A and carbon dioxide. Then this acetyl coenzyme A, in the presence of oxaloacetic acid, acetyl coenzyme A and oxaloacetic acid, which having the 4 carbon, in the presence of the enzyme citric acid synthetase, it gives the citric acid, carbon 6. Here 6 carbon atoms. This citric acid is converted into the aconitase. Aconitase is the enzyme. Citric acid, in the presence of the enzyme, biological catalyst, aconitase, converted into the cis-aconitic acids. Here carbon atom is 6. So in this way, the reaction, this cycle is continuous. Here the syllabus, as per the syllabus we are studying. You should, all of you should have the syllabus. तुमच्याकडे प्रत्येकाकडे syllabus पाहिजे आहे. So how this cycle are completing, I will show you. So here the limitation of the board, and we are conducting the lectures, work from home. So adjustment is done, the online lectures. So here the cis-aconitic acid, 6 carbon atom is formed. Then cis-aconitic acid is converted into isocitric acid in the presence of the enzyme aconitase. Here the cycle is shown. With the help of chart or by writing on the tape, on the board, I can explain very well. But due to the corona disease, we have to adapt. We have to do the adjustment. So that is a special character of the successful animals. The animals, those not adapted, they are finished. They go out of the flow. So you also try to adapt a condition, to adjust with the condition. So here the cyclic form or the citric acid cycle is shown on this chart. So cis-aconitic acid is converted into the isocitric acid. Then isocitric acid converted into the oxalosuccinic acid. Then oxalosuccinic acid is converted into the alpha ketoglutaric acid. Then alpha ketoglutaric acid is converted into the succinyl coenzyme A. Here 1 carbon atom is less. Then carbon atom is the 5 carbon atoms. Then succinyl coenzyme A is converted into the succinic acid. So this succinic acid having the 4 carbon atoms. Then this succinic acid is converted into the fumaric acid. So carbon is the 4. Then fumaric acid is converted into the malic acid, and then malic acid is converted into the again oxaloacetic acid. So this cycle is continuous. This is in the form of cycle. So it is called as the cycle, citric acid cycle or TCA cycle or Kreb cycle. So after completing one cycle, so the energy, net gain of energy is in the form of ATP, and it is 24 ATPs. So this is continuous in the body. This cycle, biological process and different reactions in the presence of different types of the biological catalyst, enzymes, it completes. So here I will send the notes on WhatsApp group in the PDF form file. So you read that, and our examination is in the form of MCQ. 4 options are there. So you have to read sentence, each and every sentence, and you also make the MCQs. I will also send the MCQ bank, question banks. After completing this unit, I will send 2 questions. One question for test first and another question for the tutorial first. So you bring the notebooks, or you can simply prepare notebook at home, but attach the first covering paper, first paper, Xerox paper. On that there is your details, your class, your roll number, your paper number, then each and everything. Test first, test second, tutorial first, tutorial second, all things are on that test tutorials original notebook. So you can Xerox that paper, first paper, covering paper, and attach with the ruling papers, and you can make the notebook at home. And in that notebook you have to write test first, test second, tutorial first, tutorial second. So in that notebook, first you write the test first, then you write the tutorial first, then you write test second, then write the tutorial second. So there should be 2 tests and 2 tutorials for the intermediate assessment for the 10 marks. So for today it is sufficient. Thank you very much. Dhanyavad.